I'll start. My name is Jackson. I'm a microbial ecologist and a geobiologist at Maggie Osborne. I'm a second-year PhD student with her, and I'm going to be talking about understanding deep subsurface microbial ecosystems and aquifers in the Paradox Basin, which is a foreland basin, an asymmetric foreland basin on the western slope of the Colorado Rockies that extends into Utah, kind of a stone's throw from where I grew up on the other side of the Rockies in the Front Range. So most broadly, our research is concerned with understanding the main drivers in microbial ecosystems that exist in the subsurface and how they influence biochemistry both in the region and the implications that that might have for global biogeochemistry, specifically in both the past and in our current Earth systems. We have a limited understanding of how terrestrial deep subsurface ecology varies across depths, chemical variants, and so on, despite the fact that the subsurface is the largest reservoir of microbial biomass on Earth. So we're kind of running into this conflict where there's a large amount of life and a large variety of life that exists underneath our feet, but we have very little understanding of how much influence they have on Earth's global biogeochemistry and biochemical systems. This problem is kind of compounded, in my opinion, because microbial metabolism mediates a lot of the chemistry that's important for maintaining life on Earth and climate during time. So we're thinking about the cycling of nutrients like carbon, sulfur, and nitrogen, and the impact that we know they have in surface ecological systems, and how those might translate to the subsurface, again, both through time and in the current Earth ecological setting. And so we're studying one very well constrained hydrological setting to help us answer this question, which is the Paradox Basin, where we know the age of the water, some of the chemical context of the water, and some of its burial and exhumation history, to kind of apply these principles more broadly. And so this is some of our sampling. We sample from like seeps that come up from the earth as close as we can before they turn into a stream, from wells, water wells, filter that water and extract DNA to help us identify who is in these systems and putatively what they may be doing in those systems. And so I can generate it in the late summer, these road maps, and this mean relative abundance bar chart is very messy and contains a lot of information, but it can kind of give us insight, like I said, a roadmap into where we can go next and who we can start identifying as important players in these microbial ecosystems, and how related these systems are to one another, and start to generate hypotheses about the relatedness and what is happening, again, putatively in the systems in this picture of the diatom. I just wanted to plug ComSciCon really quick, which is a thing a few of us in the department did, where we were teaching students how to communicate science using art and other forms of media. If anybody's interested in that kind of work, please hit me up, because we're looking to organize it again next year, and it's a cool thing we did this summer. So yeah, I think that's the time, and I'll take a question.
So you said the hydrological setting is important and also well known, so that's great. But is there a role played by the geological setting or structural geological setting, tectonic setting of the fact that this is a foreland basin? How does that affect the current microbes?
Yeah. So the primary thing I can think of to answer that question would be something like sterilization. So if we're thinking about like denudation of the Colorado River, removing sediment and bringing some of the buried sediment closer to the surface, we're talking about temperature changes through time, and so burial, but also uplift of sediment through tectonics as well is going to change like temperature and the actual like chemical composition of some of these waters. And so sterilization, I think, is a primarily important thing to think about, because if you're sterilizing an entire like water system by raising it to a temperature that's above like 120 degrees that can't sustain life, and then all of a sudden that's cooling down again, and then you're getting water that's coming in from recharge from like the La Salle or something like that. That water is bringing new microbes that are doing or performing new functions that might change how we understand their role in that system. That's the thing that comes to my head first with that question. But yeah.